Hi, welcome to Consider This. On behalf of my co-host Heidi Beidinger and myself, Marianne McDowell, we're really glad you could join us. The purpose of this seminar series, or webinar series, if you will, is to simplify the COVID-19 conversation. So we will provide some short videos and post them on the website that unpack the scientific concepts behind what we're going to talk about. And today is the first one of these. So we often talk about being infected with a bug. I have a stomach bug, but pathogens really aren't bugs. And in fact, if you look at all the insects, not all insects are truly considered bugs. There's a subset that are called the true bugs. But that's kind of a side note. There are three basic types of pathogens: viruses, bacteria, and parasites, and they differ fundamentally. So the viruses are considered to not be living, and it's just genetic material, DNA or RNA, within a particle. And these viruses must invade host cells to make more copies of themselves and to gain energy. And they hijack that host cell machinery to reproduce, essentially. Viruses infect everything. They infect humans, animals, plants, and even bacteria. And there's probably gajillions of types of viruses out there that we haven't even yet discovered. Bacteria are a little different. They're considered living. They're single-celled organisms, and they're contained within a cell wall. Importantly, they have their own components for reproduction and for getting energy, although sometimes they do live within other cells. Importantly, they have no nucleus or organelles. And so our cells contain our genetic material in a nucleus. And bacteria do not have that. And by definition, that term for that is prokaryotic. So bacteria you might be familiar with are Streptococcus, that causes strep throat, or E. coli that causes the recall of our spinach and different things, hamburgers, if you will. Only 1% of bacteria cause disease, and there's bacteria everywhere. In fact, even in our own bodies, we have more bacterial cells than human cells. We're just one big walking bacteria, I guess. So the parasites differ from these other two in that they have a plasma membrane and they have membrane-bound organelles and a nucleus in which we have our genetic material. And this is termed eukaryotic as opposed to prokaryotic. Eukaryotic cells are our cells. Our cells are also eukaryotic. So parasites can be either single-celled, like this Leishmania parasite shown here. I work on Leishmania. Another example would be the parasite that causes malaria, single-celled, or multi-celled, like parasitic worms. Most of these pathogens are microscopic in that you need a microscope to see them, but some can be macroscopic, like parasitic worms. Hopefully this hasn't happened to you. If you had to deworm your dog, for example, sometimes you can see little worms swimming around in their poop. So even though I make these, I showed these all to be about the same size. In fact, they're actually quite a bit different in size. So in general, not always, but in general, you can think of the parasites being larger than the bacteria and the viruses being very small. So this is a series on viruses. So I want to give you a little introduction to the coronaviruses. So the coronaviruses are a group of viruses. I'm only showing you the ones that cause infections in humans. And I stole this figure from this manuscript here. I'll post the PowerPoint so you can have access to it. And so here's our SARS-CoV-2. And it's a 2 because we previously identified SARS-CoV in 2003. And so SARS stands for Severe Acute Respiratory Syndrome, and it causes ARDS, or Acute Respiratory Distress Syndrome. And about 8,000 people were infected with about a 10% death rate. About 10 years later, MERS was discovered and and that stands for Middle East Respiratory Syndrome. And it was even more deadly, less infected, but about the same numbers of deaths. So there are 4 other coronaviruses that cause infection in humans. They cause really mild cold, and they're circulating all the time. In fact, I think by the age of 20, we've been exposed to them several times, and almost all of us probably have antibodies to these. These viruses are thought to have risen from animals, this group here from bats, and this group here from mice, and then get transferred to humans via most often another animal, like the camel in the case of MERS. So there's a different way of describing or grouping different viruses, and that's by their genetic code or how closely their genes are related. And so these are called the alpha coronaviruses, and these are called the beta coronaviruses. But all of them have a similar structure. And so they're given the name coronaviruses because of their spike protein that looks like a crown. And they're all a member of the same family. So one thing to point out is the virus name is SARS-CoV-2, SARS for coronavirus 2, or severe acute respiratory syndrome coronavirus 2. And that's the name of the virus. The name of the disease is COVID. So really you don't get infected with COVID, although a lot of us will use that terminology. COVID stands for Coronavirus Disease 2019. So all coronaviruses have RNA as their genetic material. They're large, about 30,000 nucleotides. So a nucleotide, or the ACTG that you're familiar of when you think of DNA, and it's also present in RNA. And so this is about double the size as the influenza virus. Coronaviruses are enveloped in that they have an envelope that contains all of that genetic material. So now I'm just going to name a few of the parts because they become important for some of our discussion later. So here's our RNA shown in gray. The RNA is covered by a nucleocapsid protein, and both RNA and nucleocapsid are things that we test for when we're testing for infection. There's the lipid bilayer, or the membrane. Some other proteins in the membrane are the M protein and the E protein, and of course the infamous spike protein, to which most of our vaccines are trying to get an immune response to. So to understand some of the concepts of generating vaccines or treatments, I think it's important to understand how we go from our genetic material, DNA or RNA, to a whole virus or to a whole human cell. So I have this little tutorial on how to build a cell. So basically you start with the blueprint, or the DNA. DNA gets transcribed or copied. This is double-stranded, 2 strands that bind together. Gets transcribed into a single strand RNA, which eventually gets read and turned into protein. So you can think of this as a blueprint building a house. The architect has a whole blueprint, and then that architect would copy parts of that and give it to different construction workers. So you can imagine this RNA is the directions for building the structure of the bricks, if you will. And so in this case, gene 1 for the human cell gives rise to this red protein. And that could be a certain component of the cell or the actual structure. And if you think of the building the house analogy, it could be the bricks. And then you could have gene 2 that gives rise to another structure within the cell, or it could be the furnace, if you will, in the house example. Gene 3, different components, could be the windows. Gene 4, it could be transporters or the door. So you kind of get the idea. It's not exactly right because Each one of these structures in here is probably made up of lots of different kinds of proteins, similar to the house analogy. But I think you get the gist. So these terms are going to come up over and over. So RNA, actually, we already did. SARS-CoV-2 is an RNA virus, but we have some vaccines that are RNA viruses, and we also use PCR to test for particular RNA. And in terms of protein, we have that spike protein, which is considered an antigen. So we'll talk and unpack all of these in later episodes. So the last item I'd like to get across is how SARS-CoV infects our cells and what happens. So here's a host cell, one of our cells. Here's our cytoplasm. That's really all the stuff in the inside of the cell. And here's the nucleus, one of the membrane-bound organelles. There's other ones in here that I'm not showing for simplicity. So we have the SARS-CoV-2 virus, and it interacts with a receptor on the surface of the host cells called ACE2. And that mediates entry into the cell. So it actually brings it into the cell and puts it in this little sac. That sac will move and change and mature inside the cell. Eventually, a host enzyme, so a host protein, will cleave the spike protein. And what that does is mediates fusion of the membrane with the membrane of the vacuole. And so the RNA actually gets put into the cytoplasm. That's where the virus hijacks the host cell machinery and actually reads that RNA. So it reads it, and then it makes the viral proteins, the spike protein, the M protein, the E protein, all of the components that the virus will need to make one of those particular particles. And it then goes through the secretory, or goes through part of the other machinery of the cell to gain outside. In addition, the RNA gets replicated and eventually gets transported to where all of those proteins are, and the virus gets made and then goes out of the cell and infects other viruses. So that's the general outline of the life cycle. So those of you that are aficionados or experts, sometimes when I describe things in a simple way, it might not be exactly right. So I'd love for you, or if I ever say anything wrong, please email us at consider@nd.edu. We will also provide links that explain things in more glorifying detail. So anyway, thanks for joining us, and I'm looking forward to the next 11 sessions. Bye.